Bonjour tout le monde, merci beaucoup de vous être connectés presque à l'heure, c'est formidable. 52 personnes à 13h32, ça veut dire que vous êtes tous en forme après les vacances. Donc j'espère qu'elles étaient bonnes et que la rentrée était bonne. On continue donc l'exploration 2025 des biomarqueurs dans les maladies mentales. Et je vais présider cette conférence avec Boris Chaumette. Je ferai l'introduction et Boris fera la partie discussion. So I turn to English. It's my great pleasure and a great honor to introduce Professor Ole Andreasen, who is one of the most leading figure in the field of psychiatry, not to say that he's the leader of psychiatry genetic worldwide. So we're very happy that he has accepted to give this talk today. Professor Andreasen is a psychiatrist and a neuroscientist, and he's currently professor at the University of Oslo, where he's head of a major initiative, which is the Norwegian Center for Mental Disorders. He has made some very important contributions to the field, enabling us to understand much better the genetic architecture of nearly all mental disorders, including schizophrenia, bipolar disorder, and depression. And as you all know, he has played a major role in building a very large-scale international collaboration, which is called the Psychiatry Genomics Consortium, PGC, to which we participate and share data. And this consortium has enabled to identify hundreds of genetic risk loci and also helped us to much better understand the pathways underpinning and underlying psychiatric disorders. On top of that, Ole Andreasen also works in brain imaging, clinical epidemiology, and precision psychiatry. And in precision psychiatry, which is one of the focus of the foundation today, he helped the field to reshape the way we think about mental disorders, moving from categorical entities towards biologically informed dimensions. The number of publications he's the author or co-author is amazing. He's has published extensively in top journals with more than 1,000 international publications and an H factor above 140. So today he has accepted to share his latest perspective on the genetics of mental disorders, and he will show, I hope, how this field will transform clinical care in the years to come. So, Ole, thank you very, very much. I know that you're in Japan at the ISPD meeting, and it's the night for you. So, thank you very much to spend this evening with us and to give a lecture on this such important topic. So, the floor is yours.
Thank you, Marianne. And I'm very happy to be here, and thanks for the invitation. I think it is really important to make sure that genetic findings are relevant for clinicians. And I know there are many clinicians on this call. I also follow the great initiative you have, and I really think you're doing a great job and have a lot of opportunities. But I also see patients every week, so I think it is if the work we're doing doesn't make any sense to clinicians, we have a problem. But still, we need also to dive into the details. And I also, these are my conflicts. I do some clinical trials also and speak to these drug companies. But the concept that we are trying to deal with is the challenges that the diagnosis overlap and the clinical manifestations. If a patient comes to you and there's always the spectrum of diagnosis and the spectrum, sorry, of phenotypes from appetite to delusions and cognition, and they could be spread out across different diagnostic categories. Then there's a lot of overlapping diagnosis. I don't need to tell you this, but just depression, anxiety is like 55%. This is American data, but this is Danish data, the registries, and there's a lot of enrichment in the overlap with the medical conditions or somatic diseases. And then another challenge is the small brain abnormalities. These are just, as Marion said, we are also working a bit in this Enigma the consortium bringing together brain imaging, also a very good group from France, and there's no major damages to the brain, so it's more like a minor small effects across the brain, as you see here, most schizophrenia, but there are these effects across many. But I would say the main challenge is that our diseases are prototypical human. That is very hard to find a mouse with delusions or a rat with attention deficits. So this is the background for why we are using genetics as a tool to understand better the underlying phenomenon and see both if we can understand more of the mechanisms, how genes interplay with environmental factors, and then also if this can be brought into the clinic. So I will start with the current status, so try to do it briefly about just the genetic discoveries, then talk a bit about the mechanisms, and then finally, what are the potential for clinical translation? And this is the PGC, as Marion said, it's a big international kind of discovery factory for genetic variants. And it can do two things, like biological insights, maybe drug targets that it could be useful for new drugs in psychiatry. And then could it be used for precision medicine tools? And I will focus mostly on the translation part on these precision medicine tools. And then we're also doing this non-European trans ancestry analysis as well. And now it's so cheap to genotype your DNA. They say in the US or also in Norway, it's cheaper than parking at the hospital to do the genotyping, and this is technology driven. And I can show you that this has allowed us to genotype like hundreds, thousands of samples. And what is really clear is there are very many small effects. It's called polygenic architecture. It's not like, oh, here we found schizophrenia gene. Oh, this is a bipolar gene. So it is really these polygenic effects. And how do you do this? You can do this genome-wide association studies. It's not like favorite gene anymore. It's just screening the whole genome. It's called GWAS, genome-wide association studies. And you compare cases with controls. And for each of the individuals, there are like 2-3 million variants. So this is a discovery. It's data-driven. It's not something you do on your laptop at your kind of lab. This is big data, a lot of servers, high computing systems, high performance computing. So it is turned into like a big data approach. And this is, the recent paper in schizophrenia, and each of these variants in the genome, these are all the chromosome. We have 22, and these are all the variants of each chromosome, and the higher the y-axis dot here, the more significant it is. So this is just p-values. And you can see there are a lot of significant p-values. Many, many, many variants on the genome have an effect associated with schizophrenia, and this is 10 to the minus 42. So that's why, this is highly, highly, highly unlikely to be a false positive, for example. And then you can do the same in Alzheimer's disease, 75 loci. Those of you who are into genetics, ApoE has been on a long time, but now you can see also this kind of mixed polygenic effect, many, many variants across the genome, also associated with Alzheimer's disease. And then major depression, that's really, really we now have the most variants. This is 635 loci. But what is the case is that to find these small effects, you need big, big samples. And here, there are more than 500,000 cases and more than 2 million controls. So this is huge data being used. And then if you need to have millions of participants to find anything, you start wondering, I'm seeing one patient. How can I use this information for the patient in front of me? And that's the same with bipolar disorder. This is Kevin O'Connell who led this work, came out now recently, contributions from France as well. And here, 160,000 cases. So it's massive. And here we have 2.7 million. And there are now 298 loci all over the genome. And as you can see, there are significant effects on many, many parts of the genome. And this is only a statistical association. We have to investigate this close to figure out what's the thing. And then there's also, you see here, we added like 236,000 from Latino background, East Asian and also African. But in East Asian, there were specific loci not found in European samples. Anyway, and here you can see this is what I found a bit interesting with this because the way we do recruitment in these big studies is we can go to hospitals, various clinical assessment. You have SCID, you have these very highly structured tools, or you can go to the community, which is here mainly healthcare data from Scandinavian registries or biobanks from the hospital. So then there is not this specific what do you call it, screening, highly scientific, but it is done by professionals. And the good thing here is you have a wider variation. Kind of here you can capture more of the whole picture in psychiatry. And then this is also self-reports. This is from this 23andMe, where people say yes, no, do you have a diagnosis? And totally there are 158,000, 2.8 million controls. And that's what we found. And so how can this now be translated? Remember, this is called p-values. Everything else so far is just statistical genetic analysis. Sorry if my internet is... So bipolar disorder, then we could extract the credible is very focus on the whole genome with genes, because the genes can then these mechanisms are involved or not. And I just want to highlight some of them, like CACNA1B, that is calcium, which makes a lot of sense for bipolar disorder. In they another type of find 70 publications and just so many genes there, so that of information here, and we can do fast to find will X, but this is more like a annotate to the genes. depression somewhat. This another way genes they found like three genes. They were mostly receptive synaptically. So these were the genes. So now I know more about specific gene. It's three. So right, and then schizophrenia, the severe mental disorder. Then what can this the calcium seems to be there are calcium related molecular functions involved, and this yes, it's okay. Thanks. So yeah, back to the calcium channels, and this is something that's been repeatedly found now in bipolar disorder. So, and that is really something, you know, it's possible now to run maybe clinical trials with the calcium blockers, but I think they've even been tried in some pilot studies. But that's definitely something that could be tested out now. At least there is some genetic evidence for it that that could be a pathway and could be also a mechanism for bipolar disorder. Anyway, then where in the brain do we have these findings? And now it's also possible, that's why genetics is an interesting tool because we can, if we find something in these big studies, we can take the genetic variants and link it up to post-mortem brain. And it's not possible to do all kind of sequencing of single neurons in the brain. And this is just to show that this is enriched in different regions. This is in cortex. This is in median spiny neurons, pyramidal. So it's mostly in hippocampus and in interneurons in the prefrontal cortex. So this seems to align with ideas or post-mortem data already known in bipolar. Then uh, then you can do this single neuron. You can go look at this. There seems to be all the variants in bipolar seems to be enriched in specific regions of the brain, and it's amygdala, underscoring the mood kind of focus of bipolar, something about the intercephalic regions, hippocampus, and then some interneurons. So this is kind of using the discoveries from these crazy big samples, variants associated with different regions of the brain. And then finally, so it's the GABAergic interneurons and it's median spiny neurons. Then you have is another way of looking at it, how you can do this analysis. And what is interesting also is that there's not only the brain, and I think it would be strange if the brain was not involved in genetics of bipolar disorder, but even after controlling for everything was happening in the brain, there is, it's called conditional analysis. There is a signal in enteroendocrine cells and also in the large intestine, these delta cells in the pancreas. So it is quite interesting that, much higher signal, of course, in the brain, but then you have some effects in the gut. And this also speaks to this comorbidity that we talked about in the beginning. And then just finally, there was a new paper. So what I've discussed so far is the common variants, but there also now there's another finding here from the Decode guys also came out this year that these are rare variants, very, very rare. It's like only 10 people in Iceland having them. So it's hard to use it as any kind of clinical, but this is something where you can use for kind of new drug development. And as you see here, both of these genes that are significant, the ACAP11 and HECT-D2, interact with this, remember the GSK3 beta? That's definitely shown that lithium inhibits this. So it could be a mechanisms here that could help us with new drug development in the future. And then just something about schizophrenia, the same point there. He just they took all the variants found, did this kind of polygenic enrichment into cellular regions of the brain. And here you can see where those are the strongest signal. It is in the frontal cortex, something also here in the accumbens, amygdala, the mood part of the schizophrenia kind of phenotype. And then the anterior cingulate, and then of course there are all over the cortex. So there are, this also recently came out, and they can also link it to specific neurons. So now I think going from these massive p-values that are just statistical association, but linking that to biology could guide a lot of the follow-up neuroscience studies to find new drug targets. And there they also had, I don't know if you remember, but schizophrenia and all the psychiatric genetics was never replicated. But here you can see the larger the samples, it was from 2011, then 2014. Then you can see a clear pattern of enrichment in these tissues, is frontal cortex, cortex, nucleus accumbens. And it's the same now in this more recent paper. So find that we have signals for the biology of schizophrenia that's clearly replicating, just like in bipolar, where we had this, for example, the CACNA finding, this calcium channel. But then it's also they link it up to synaptic genes. All these prioritized genes. They seem to be in many of them seems to be in linking up in the synapse machinery. Those of you who know this better, but you can see these genes, how they are linked to kind of synaptic localization, these prioritized genes that I remember I talked about earlier. And then again, they have this rare variance here also. So you can see that seems to be some of these rare variants also that are associated with schizophrenia. They are also now becoming more and more. There's some technical terms here, but I'm just mentioning this because there are also these rare variants that are associated with schizophrenia, not only the copy number variants. And this seems also to be the point in this paper was that the genetics seems to be same across different populations, African American, East Asian Europeans. So you can see that it's pretty nice, that there's larger variation in these other population Europeans. They are the most larger samples, but then you can see that it is much less variation. And this seems to suggest there's something with the kind of the integration of this neurotransmission. There's some kind of called fast and slow. This is the Greengard paper, Nobel Prize winner. Then we just linked all of these new genes to the cellular machinery, also intercellular, who is working on maintaining homeostasis in neurons. So there's quite a lot of exciting things happening to understand now the machinery of schizophrenia. And the same with major depression. You see the same patterns here. Here, the smaller sample, they don't have the same effect sizes, but now you can see that the different types of neurons that the genetics are enriched in seems now to be replicating, and you have a nice pattern of findings that are definite for for depression. And then they didn't have that kind of strong findings, so they also did something so they called it gene ontology synaptic, and there seems also here to be uh, kind of synaptic postsynaptic mechanisms in depression. And they also, or I was also on this involved in this, so we also tried to find new variants, new drug targets that could support the repurposing of known drugs. And here you can see an overview. There are some of these that could have an effect on the, or be implicated by the genes associated. Here you can see the number of genes associated with the drug targets. So then I just want to spend the last quarter then on clinical translation. So I mentioned the potential for drug discovery, but how can you bring this into precision medicine? And take out the cancer or tumor, do sequencing of the tumor and find the variants that they then can say, oh, here we have this variant. Then you can select this treatment because the variant is really linked up to mechanisms. We don't have that in psychiatry yet, or we'll never have this kind of sequencing, of course, and we can't go in and do anything in the brain, of course. But I think we need then still to think about this precision medicine. So genetics is one thing, but I also think, utilizing health records, they're becoming more and more electronic now. Phenomics, in recording from apps or passive sensors, and then big data. This is going to really be important in our field and artificial intelligence. And I think this is one thing is with discovery when we do this big studies, but another thing is how can this be translated to the clinic? And this is what we are working a lot on now. But then if you want to have do precision medicine, people need to trust you. It's never ever going to happen if there is lack of trust. And then you also need large samples to follow people over time, and it has to be diversity. If you only have Scandinavians in your studies, it's not going to be really valuable because you don't know if this is only something happening in Iceland or 350,000 people, or is it something that's also seen among the 6 billion other people on Earth, where maybe it's even more now. By the way, so this was written by the previous head of NIH, Francis Collins. All that some other people running the show there, but still, this was written to plan. This is the focus of what will happen in 2030 in healthcare. But I think they never mentioned psychiatry, but this precision medicine using what we have, not only genetics. I'm just genetic, that's why I do it as a research, but we should also use these other tools to make personalized treatments. And we can start already now. You don't need to wait for some genetic discoveries, but set up this system and make it more clinically relevant. But I will talk a bit about what, oops, here is something happening. What can happen with these kind of personalized trajectories. So if you go have a patient, do all kind of assessment, clinical imaging, kinetics, put into like a big data, machine learning, or even artificial intelligence, and you can build your digital twin, and then you can follow the trajectory and see much earlier if you go not under what is the norm in the population, but when you start to deviate from your personal trajectory. And that's what we really are focusing on. And then this is what can happen in when you do this polygenic scoring. You may have heard about this, but one thing is when we did it with all the brain cells and everything, but we can also do it in patients. So it's called the cumulative effects of all genetic variants, because each of them explain next to nothing of the variance. But maybe if you do combine them, they will have larger effect. So you can then, if you have a genotyped individual, you can calculate kind of the risk of disease. And this is used a lot in schizophrenia. Here are the liabilities, how much is it explained, but it's not super high, as you can see. It's maybe getting up there to, 10, like liability explained 10%. So it's not really, really, even though with all these variants, it's just polygenic risk scoring is not helpful for predicting schizophrenia. So, but the concept is there that you can have explain more of the variation in the population. In bipolar disorder, you can see the same pattern, but here's a bit higher, like in bipolar 1. We're up now on like 20%. But then if you do include the self-report in your sample, it's quite lower. So that's much more unspecific. But if you go here to the right on the community, then you can see we're getting up there to 25%. Of course, here is a large variation, so we can't use it for clinic, but just the concept that you're getting up there now, or the potential that you can have explained variance around 25% just by doing a genotype is the concept is interesting. And then in depression, you can see that it is even lower. So depression is less heritable, even though we have so many hits. The highest explained variance is kind of 6% here. And you can see there's some differences in how much the odds ratio. But then what you could do then is, now I'm into this big data, so you can sort of plug this in. If you have 2 million people with 5 million variants each, we can then, and this is just plotting every variant in bipolar disorder and the effect sizes on the y-axis for bipolar, depression effect sizes on the x-axis, and then the red is the overlap. And then we can estimate this based on the like mathematical modeling, and then you can figure out, look, these are all the variants in bipolar disorder. You can then get the number is should be 7, like 14,000 variants all of the genome, not only those that are significant now, but just estimate how much of the genome. In bipolar, it's 8.6, and there is like 7.5 thousand times 7,500. That's overlapping between these. This is based on somewhat of a genetic overlap estimate. And if you do those variants here, you see the overlap inside here is much higher. This is some kind of a estimate for that. But overall, the correlation between bipolar and schizophrenia is lower. But then you could think that these variants that are out here, not shared with depression, is involved in manic episode. But that's something we're working on to figure out now. And then you can see there's a lot of overlap with the schizophrenia and bipolar. That's even higher here in bipolar and then this is the bipolar and ADHD. Also here a large overlap. There's a lot of overlapping symptomatology. And here I don't want to go into details, but you can see that the genetic architecture is, if you focus on this top left here, the clinical and community samples are quite the same genetic architecture, but the self-reports are a bit outside. This is clinical, then self-reported is a bit outside. And then you can see that bipolar 1 and bipolar 2, they are outside. There are many variants not seen in bipolar 1, but all the ones with bipolar 1 are also seen in bipolar 2. But bipolar 1 is mostly overlapping also with the community, and then the bipolar 2 also in the community. But you see the self-report are mainly they're consisting of bipolar 2, but of people not seen or record or recruited from hospital settings, but they're much smaller overlap with bipolar 1. So this seems to be the self-report more of a, what do you call it, lower symptom load, the bipolar phenotype. And here you can just see that there's a lot of overlap between, this is the genetic correlation between schizophrenia and the different types of bipolar disorder, and that there are more overlap also with PTSD and also with ADHD across, and some also with personality. And there are different patterns depending on which samples you are looking into. So as you see, there are more linked to ADHD and PTSD, and those with self-report. So this seems to indicate that there are more overlapping phenotype, not so stringent um, clinical assessments, or there could be more comorbidity in people reporting or not being recruited at the hospital. And then this thing about spending hours talking about the genes and all the bad things and everything, but I think it is quite important to remember then that there's a quite strong genetic overlap between creativity and both schizophrenia and bipolar. And on the left hand here, you can see the polygenic risk score, and that predicts the creativity in the population. Not very high, but quite very significant. There are also some indications of improved cognition in bipolar disorder. And this concept that bipolar disorder is specifically, is associated with creativity, artistic temperament, I think that's quite interesting, both from an evolutionary perspective, but also, that having, for example, bipolar disorder there are also some of the disease that could be, what do you say, advantageous or linked to advantageous phenotypes that at least the human beings as a society, that's what we need to move forward. And then just the final, I mentioned Alzheimer. I know you're not working so much on that, but you can imagine how this, if it's possible to use these new tools in the clinic. And I think this is the best example we have so far, that for Alzheimer, you need to start treatment before, the brain is damaged. And who should be selected for intervention? Now we have the new drugs. And then we have developed some scores that we could, you can combine both imaging and genetics and clinic and biomarkers, and you can then add imaging to this. And this is, you can develop, this is some kind of a machine learning, so capture variants in the brain that's most associated with dementia. And combining this, there's something here. Well, anyway, the figure is like this. This is machine learning, or kind of that you have the probability on the y-axis here. Nobody have dementia at baseline. You come to the clinic and say, I have memory complaints. And then if you do this genetic, the polygenic score, and you can see that based on the risk percentile, that there are differences in the probability of developing 5 years Alzheimer. So these in the dark here, most of them will develop up here. There seems to be more other issues that can explain your memory complaint. But this is, odds ratio 3.5 in the clinic. Forget that this is not clinically useful. So genetics alone, not helpful. Okay, but then let's put another feature into the model, which is this imaging score, imaging hazard score. And now you can see it's Now here again, this is everything is based on data collected at baseline because you do imaging, often or should do in the memory clinics. And you can see now the guys that are not developing, they sort of s- getting up there, and these groups here, very little likelihood, but here is more, you have the patterns of these that will develop, and now the odds ratio is 13, and then we're talking clinically relevant. But then finally, what you also do in a memory clinic, day one, is to do cognitive testing, and this is based on just regular clinical assessments. It's called a verbal learning test, and here we can see now there's like two groups now. These groups here don't develop Alzheimer after 5 years. And down here, these are pretty sure you will develop Alzheimer. And this is the type of groups that should be selected for treatment, because here you will only have the side effects. It's not going to help you if you start treating people early without specific diagnosis. And this can also help you with reducing the sample size if you're interested in running clinical trials. This is something as you're moving into that phase now, you can think about. So if you do all of these assessment, then the sample size for a clinical trial is reduced to 33%. So it's a lot of cost savings to do this kind of clinical trial assessments. And this is something then we could put, this is the future of precision medicine. Can we put all of these together in genetics, biomarkers, cognitive testing, imaging, and put them into like a dashboard? I think I showed you this earlier also, and get people involved to understand kind of risk or management of when you will develop the disease or a phenotype. And I think this is something we're testing out now in the real world life to see if we can get early markers. But this also, we're doing the same in bipolar, it's not so impressive the numbers here, but if you do genetics alone, this area under the curve is 0.67. And you did all of these machine learning tools and imaging, you get up to 0.59. But combining them gets it higher. It's not magic here, it's just proof of principle. And we have higher numbers now when we have done it in the most recent. So it's absolutely possible to think about this. But then we need help from the clinicians to say, well, you want to do genetics and imaging? Maybe it's better to do SCID, and then we can get the diagnosis. So I think this is more proof of principle, but finding the right use case, the right scenario where you can assess these or use these new tools. And I think in selecting a pharmacological treatment, there we have some preliminary data that seems to be more relevant. And this is also something we may discuss. And then finally, this is not happening without the involvement of stakeholders, user groups, to figure out how this could be done. That was something we did in the ECMP, to have like a roadmap for how to involve this approach and get it into clinical practice. So I think I'll finish there. So we have now 300 to 700 these genome-wide signals for these three major disorders. There's a large genetic architecture that's overlapping, but and I would say it's promising. Others say it's totally not working, but this prediction may not be clinically relevant, but stratification, can we predict who will uh, use or have less side effects of a given drug? Or if you have a mood episode, is this bipolar disorder or is it depression? And these studies we are doing now to compare. We have this big depression and we want to run it together against bipolar to see are the genetic factors that are significantly different. And then there's a lot of new stuff now on the biology and mechanism that, in bipolar, the calcium signaling. schizophrenia, these synaptic factors, and more generally in depression, and they also seem to highlight some drug targets. So with this, I just want to thank all the hundred thousands of participants of these studies. It's quite amazing how many volunteered to be involved and make this possible, and then a lot of uh, people working on all these samples and also funding mechanisms. Thank you.
Thank you very much, Ole. It's, as usual, extremely impressive. Such an amazing amount of work. I'm going to turn the microphone to Boris, who is going to lead the discussion in the 10 last minutes. Boris, thank you.
Thank you, and thanks a lot, Professor Andreasen, for this very inspiring talk. And I'm sure there is like hundreds of questions we can ask you after this presentation. But if you want to maybe have some feedback from you on some specific points, so I was very interested by the question of the GWAS hits that are pointing towards intestine in bipolar disorders, because in my opinion, it's one of the first time that we are going outside the brain with genetics of psychiatric disorders. Do you consider that it's now a disorder of the whole body that we are dealing with?
Well, I think this is quite trustworthy. It's a robust finding that there seems to be involvement also in the intestines or in the pancreas. But it's there are lots of, all the side effects of SSRIs is because you have all these receptors in the gut. So I think there could be some commonalities there. And Marianne, you guys are working a lot on the immune factors, and I think there could be some like genetic commonalities also there. Uh, I think there are definitely some opportunities for thinking a bit new on these diseases. But before we conclude, remember it's like a couple of hits. It's a p-value, and you needed like 2.7 million controls and 160,000 to find it. So it's not like a major driver, but at least it's a conceptual finding that we shouldn't overlook it, I think. That's my point. But and there could be some interesting ways to understand better.
Thanks a lot, because I'm sure it's connecting with some ideas we have about this in the
Exactly.
group. Yeah.
Yeah.
I'm very impressed by the summary I've done from all this work, because just to let colleagues know that it's probably 20 years of research to find all these hits and to aggregate all these cohorts in the PGC, and you have led this consortium and this effort with so many colleagues in the world. So it's a very effortful initiative. And now I'm very also pleased to see that you're trying to connect with the clinical practice, and I'm sure that's connecting with what we are trying to do now. And I will just ask you a little bit about the polygenic risk score, because there is also some publication challenging the reliability of polygenic risk score in clinical practice. I'm sure you're aware of this. There is some effort in cardiovascular disorders to translate polygenic risk score, but sometimes the tools can lead to conflicting results on polygenic risk score. So, how do you approach this translation?
You should be careful, because it's just translating it without validating, without making sure it's working in real life. So, because remember, these studies are based on all kind of the assessment, how they were created. And then you suddenly, oh, we have some magic score here. Let's test it out in a clinic. It's not tested out in what setting and in what type of population. So all of this has to be validated. So it's a long way to go from like a paper in Nature with some kind of p-values and a clinically validated tool. That's one thing. And what do you call it? Garbage in, garbage out. So do you now know if the phenotypes of this 160,000 are reflected in your, is that relevant for your clinical decision? So there, and I know that, a lot of in schizophrenia, a lot of the sample is collected from these Leponex clinics. And so there's, but so we need to go back and look at the numbers. And I think also, finally, that the GWAS, that's just a case control to control for confounders to find mechanisms. That's why they discovered or do the GWASs. To do prediction, that's a totally different ballgame. So there you need, that's another mathematics. Discovery is one, but so that's why we have started doing this kind of polygenic hazard or taking age into account and those things. So I would be very careful to use the polygenic risk scoring, which is it's a byproduct that's easily available because it's not sensitive and it's published and you can use it. But that doesn't mean that it's clinically relevant. So I think we should be very careful. That just some dude doing a GWAS and you collect those samples from the web or whatever, and suddenly you have a genetic tool that you can use in a clinic. I think, but on the other hand, I think if you as clinicians could sit down, okay, what could be the most relevant clinical case? I mentioned just this, maybe side effects of antipsychotics or differentiating subgroups with immune factor, there could be, but this is something you need to talk to the analyst about because they have no clues. I can assure you. Back in the days we had an analyst and he said, "Oh yeah, I noticed this name, this terminology, schizophrenia. It comes up anywhere. What is that?" So you should. Sorry for putting it, but this is quite important to get clinical knowledge into this kind of big data analytics, and that I think. Like your team, you have a lot of opportunities here. You can take these big samples out there, find good use cases in your clinical network and figure it out what can be used and what is the best approach.
Yes, thanks a lot because it's also going in the same direction as the things we are discussing in France. For now, we did not propose to test the progenic risk score, but we are involved in the process of using whole genome sequencing in clinical practice. Now it's available for certain form of schizophrenia and bipolar disorders, especially based on the results you have with ACAP11, for instance. So we're looking more at rare variants.
Yes, but then I think you need to be careful. Why? Is it to do drug discovery? Is it for clinical? So I think, if you are afraid of multiple testing, since you have 5 million, if you do whole genome, you have 4 billion. So I think it is, you need to be very careful what could be the question. And I think finding rare variants for drug discovery, that is that is what they're doing, like Decode. That's what their business plan is, to find rare variants, take those rare variants, see if it's druggable to develop new pharmaceutical agents. But I think to do explain variation from rare variants, that they are rare. That's the point. So it's hard to do any kind of prediction based on that, since there are so few.
It's not for prediction, it's more for assessing rare disease behind psychiatric disorders that we're trying to
I
do
see,
it.
yeah.
Yeah. All right. So
That's a good point.
yeah, thank you. Maybe I can leave the floor to others if they have questions, of course. And see if somebody's raising the hand or otherwise I will just ask you another question if I may. We are collaborating together in the PGC, especially the functional genomics working group of the PGC, and developing the same kind of approach as GWAS, but for methylation. And we have a paper together in preparation with Stéphanie Lehéla. And now I saw some also interesting score coming from these epigenetic factors like methylation risk score. Do you think it's also valuable to incorporate this kind of information into a composite score?
I think take no prisoners. I have no special. If it can predict, then it's fine. If it's imaging or genetics, cognitive testing, you should. There's nothing special with genetics. So if methylation could help you, but it's. I would more phrase it as we call it the testable hypothesis. Just show me the data. We will, or do it. It's a great opportunity, but I don't know. It's what we did in this genetics, imaging, and cognition. At the end there, it didn't increase much for a lot of the genetics as already explained variants, and then imaging, so anything that could add, just use it. I think that's an
Yeah,
excellent idea.
thank you. Bruno, I saw you raise your hand.
Yeah, thank you, Boris. Thank you, Ola, for your very nice presentation. I have just a naive question. How do you explain the low level of association between several cell type, especially himself, microglia, or in several tissues, adrenal, pituitary, or hypothalamic, referring to HPA axis? Because you have a large body of literature support the role of heme signaling or stress-related pathway in mental disorders, regardless of the diagnosis made.
So I think it is the problem with genetics that it took a long time before even dopamine receptor came up. So I think it is probably it's not just tagged by the genetic variants. There could be some maybe not so much variation in that region. So it's just more it's not like polygenic, or no, it's not the minor elite frequency is maybe it's not much variation or something. So that it doesn't. It's very difficult to use genetics to rule out anything.
Hmm.
So it's hard to prove that. Yeah. So it's more like sorry that we don't have the tools to figure this out yet. And I totally agree. We pump out all these big genetic studies and try to, CACNA, whatever, but then these other things that are could be even more important, they are not up there because it's not tagged by the genetic variants. So you have a really good point.
Thanks.
Thank you. If nobody wants to ask one more question, I think I can leave the mic to Marion for the final words.
Well, just thank you, Boris. Just want to say a big, big thank you, Oli, for an amazing decade or decades, and hope to see the 20 coming ones to see more progress and more understanding and more new treatments. So thank you very much for taking the time. Enjoy your congress in Japan and hope to see you soon.